Good afternoon, all. Welcome to botany sessions. In these videos, we are going to discuss one of the 11th chapter, transport in plant. The subtopic is guttations. Then, what is called guttations? Then, what's the difference between transpiration and guttation? So, these are the things now we are going to discuss. Guttations. What do you mean by guttation? When it will happen? It compared with the transpiration. Guttation takes place in the early morning. Then what is called transpiration? Excess amount of water is lost in the form of water vapor through stomata. These are called transpiration. But transpiration occurs during daytime, high pressure, high temperature, transpiration takes place. It's happened during daytime. But whereas in guttation takes place early morning or late evening, due to high atmospheric pressure. Or humid. There is a more humidity, the rate of transpiration is much reduced. So under this situation, the water absorption of plant takes place. Due to this condition, the root pressure is developed inside the plant. Because if the large amount of water is absorbed, among the amount of water, only 1% of water is utilized by the plant for various metabolic activities such as photosynthesis, respiration. Just only 1% of water is utilized. The remaining 99% of water is lost in the form of water vapor through stomata. These are called transpirations. But in guttation, actually, water absorption also there, but the rate of transpiration is cut down or reduced, very much reduced. As a result, root pressure is developed. The root pressure is developed due to the accumulations or excess amount of water is present in the plant. Under this situation, guttations takes place. I mean, what do you mean by guttations? Excess waters that exudate in the form of liquids. Excess amount of water lost in the form of liquids, not a pure water, liquids in the form of liquids through the edge of the leaf is called guttations. But transpiration takes place through stomata, but guttation takes place through a stomata-like structure is called hydrodotes. Let us take diagram for clear understanding. It is a leaf structure. Both the leaf surface covered by a barrel-shaped parenchymal cell. These are called epidermal cells. The epidermal cells are tightly packed without any intercellular spaces. It is the outermost layer of the leaf is called epidermis. The edge of the leaf, it is a minute opening. It is covered by guard cells. There's a center portion of the leaf is midleaf. Midleaf is nothing but they form vascular bundles such as xylem and phloem. A water conducting element xylem, it is composed of tracheids. It is a major water conducting elements present in the midleaf. Both sides of the midleaf is covered with polysaid parenchymatous cells. The polysaid parenchymatous cells are tightly packed without any intercellular spaces. It contains more number of chlorophyll pigment, so they mainly involved in the photosynthesis. So these are called palisade parenchyma. Then next to the palisade parenchyma, spongy parenchyma. The spongy parenchyma, they are irregular in shapes, loosely arranged with a lot of intercellular spaces. It's a spongy parenchyma. See, there is a mass of cells. It is a mass of cell which is present over the xylem and phloem. They are loosely arranged with the intercellular spaces. These are called the epitheme cells. So once again I would remember, the guttation takes place through a minute opening. This structure is nothing but hydrodotes. Hydrodotes, it is also small opening like structure. It looks like a stomata like structure, but stomata present more on the lower epidermis than that of the upper epidermis. Gutations takes place through edge or leaf tip. It is covered with a stomata like structure is called hydrodotes. There is a space which is present over the epitheme cells. In this space, the absorbed water, all the excess amount of water is absorbed by the plant. It is accumulated in this space. Due to root pressure is developed inside the leaf, the excess water does not take space through the stomata. It lasts in the form of liquids. Liquid, it's not a pure water. It is composed of a solution that contains a lot of organic and inorganic substances or gets dissolved in it. Organic and inorganic substances gets dissolved in the form of liquids. So guttation in the form of liquid, but whereas in transpiration in the form of water vapor. There is a major difference between guttation and also transpiration. Liquids, it takes place through hydrodotes. So these are called guttation. For example of gutations, the gutations takes place certain grasses. Grass, it is a monocot. Potato, tomato, brinjal, then alocasia. Alocasia, it is a plant that belongs to the family Aeraceae family. Aeraceae family. It is commonly called as elephant ear. The leaves that look like a elephant ear-like structures. Hence, it is also called elephant ear, common name. But alocasia. So these are the example for gutations or takes place in the plants. These are more or less monocot plants, not for dicot. In monocot plants, gutations. The next topic is measurement of transpirations. Right. We know that transpiration. When it happens, transpiration happens during daytime. And how can we measure the rate of transpirations? See, by using an instrument is called Gannon's photometer. It is an apparatus which is used to measure the rate of transpirations. How can we measure the rate of transpiration? The rate of transpiration is measured by using an instrument is called Gannon's photometer. Indirectly, not for directly. This instrument was first made by Gannon's, hence it is also called Gannon's photometers. Indirectly, how can we measure indirectly? Based on their amount of water is lost in the form of transpiration, that's equal to the amount of water gets absorbed. So based on this equal amount, we can measure the rate of transpirations indirectly. So before we are going to discuss, first we want to know the structure of photometers or Gannon's photometer. The Gannon's photometer, it is an horizontal graduated tubular-like structures. It's a horizontal graduated tube. Both the end of the tubes are bent in opposite directions. How? One end bend upwards with a wider mouth. These wider mouth regions are closed with one-holed rubber cork. One-holed rubber cork through which a small twig is inserted. Small plant or small twig, the small branches of is inserted. The gap is completely sealed with wax. Another free end of the graduated tube is bent downwards, which is kept or dipped in a beaker containing colored waters. Beaker containing colored water. So beaker, colored water. So there is a water reservoir which is fixed to the horizontal tube. It's a water reservoir through which water can pour. Here the stopcock is fixed. These are called stopcock. Then what is the function of this stopcock which flow the water, which regulate the flow of waters? These are water reservoir, stopcock. So this entire physiological setup is fixed in a vertical position with the help of stand. We can fix vertical position with the help of stand. Now, this whole physiological setup is filled with water through water reservoir. If you pour the water through water reservoir, it can completely fill with water. And a small air bubble is introduced. Air bubble. A small air bubble is introduced into the graduated tube through the narrow end. One end of the graduated tube is a wide end, another end is bent downward with a narrow end. A air bubble is introduced into the graduated tube through the narrow end, and this whole physiological setup is kept in a bright sunlight for few hours. After few hours, after regular interval of time, we can measure the rate of transpiration. How can we measure the rate of transpiration? Based on their water absorption or based on their bubble distance traveled by the bubble. If the plant can transpire, water is lost in the form of water vapor, the air bubbles, they move towards the twig. So based on the distance traveled by the air bubble, we can measure how much of water is absorbed or lost by the plant through transpiration. That is equal to the amount of water is absorbed from the beakers. So based on this regular interval of time maybe 1 hour or maybe 2 hours or maybe half an hour, based on the distance how much of distance traveled by the air bubble, we can measure the rate of transpiration by using the instrument is called Gannang's photometers. Is nothing. It is a graduated tube, ball-like structures, horizontal graduated. The both ends are bent in opposite direction. One side bent upwards, another side bent downwards. So this is a way we can measure the rate of transpiration. Another method to measure the rate of transpiration, cobalt chloride. Chloride paper method. COCl2, cobalt chloride paper method. Then with the help of these strips or cobalt chloride strips, how can we measure the rate of transpirations? See, maximum the biennial plants or annual plants, the more number of stomata frequently present on the lower epidermis than that of the upper epidermis. If the number of stomata present more on the upper epidermis, what will happen? It enhancing transpirations. Due to sunlight, the stomata kept open during daytime, it lost excess waters. If the all absorbed water gets transpired, what will happen? The leaf become dry and withered and automatically shedding from the plant. The rate of photosis are arrested, automatically the plants leads to death. So to cut down the rate of transpiration, to prevent the water loss, more number of stomata present on the lower epidermis than that of the upper epidermis. So how can we prove the stomata present on the more on the lower epidermis? I mean the transpiration more on the lower epidermis than that of the upper epidermis. How can we prove? By using a strip, cobalt chloride paper methods. First, we have to select a healthy dorsal ventral leaf. This is a potted plant. Yes, it's a potted plant. You have to select the healthy dorsal ventral leaf. What do you mean by dorsal ventral leaf? If you cut the leaf, it is differentiated into upper palisade parenchyma, lower spongy parenchyma. A leaf that shows such type of differentiation is said to be dorsal ventral leaf. I mean dorsal side and ventral side. Dorsal side is upper surface, ventral is nothing but lower surface. The dorsal side have palisade parenchyma, palisade, and the ventral side have the spongy parenchyma. See, they are both type of parenchyma cells are get differentiated. Such leaf is called dorsal ventral leaf. It is a characteristic of dicot plant. But whereas in monocot plant only either palisade or spongy, but never both. Such leaves are called isobilateral leaf. It is a characteristic of monocot. You have to select a dorsal ventral leaf. These are the dorsal ventral leaf selected, and this both the upper and the lower surface of the leaves are cleaned with the dry cotton. It's the first procedure, dry cotton, and place there a blue color strip, cobalt chloride strip. Yes. It is a cobalt chloride strip. The cobalt chloride strip actually it is a blue in color. It is placed on both the surface of the leaf, both the surface, and immediately these strips or paper is immediately covered with the glass. I mean glass slide, which make them to immobilize. The strips they cannot move by placing this slide or glass slide. If we place the glass slide over the cobalt chloride paper, it makes them immobilized, and with the help of sticks which tied over that. Now this full experiment, entire experiment is kept in a scorched sunlight, I mean extreme sunlight, for few hours. After few hours, what we can observe, especially the lower surface of the cobalt chloride paper, which turn into pink color. Pink color. Here what will happen? The strip actually is a blue color, but due to hydrations, the cobalt chloride papers become hydrated. COCl2 to H2O. Hydrated, or maybe 4 H2O gets hydrated, which turn the blue color into pink color. That indicate that more amount of water vapor is released or lost in the lower surface than that of the upper surface. So we can clear that the more number of water vapor is lost lower surface than that of the upper surface, because number of stomata more on the lower epidermis than that of the upper epidermis. That is why the lower surface of the strips become turned into pink color. So from that experiment we can conclude rate of transpiration more on the lower epidermis of the leaf than that of the upper epidermis of the leaf through cobalt chloride paper methods. It is another method to measure the rate of transpiration. The next, what are the importance or significance of transpiration? Yes, already I point out, nearly 90% of water is lost in the form of water vapor through stomata. The total absorbed water by the root system, only 1% of water is utilized for various metabolic activities. Remaining 95 and 95% of water is lost through stomata by the process called stomatal transpirations. If the 95% of water is lost, it seems to be evil to the plant. What do you mean by evil? It is harmful to the plant. If the transpiration is continuous for few days, what will happen? All the absorbed water is lost, it leads to death of the plant. So it is a evil or seems to be a evil to the plant. It was first stated by Curtis. So Curtis stated that transpiration is a necessary evil. It is a necessary evil to the plant. Stated by Curtis. Necessary, yes, of course it's a necessary. A lot of benefits is also there. At the same time, it is harmful to the plants. What are the necessary? However, a lot of process, a number of process, for example, absorption of water, ascent of air, absorption of mineral absorptions. So these are the number of process which depends upon the transpirations. How? Water absorption, especially where the soil is a source of water, soil is the source of waters. But the soil water is absorbed by the root system due to transpiration pull. If the water is lost, it creates a pull that is called transpiration pull. Due to transpiration pull, the water absorption takes place. It is only accepted theory, transpiration pull theory only accepted. A lot of theory was put forward to explain many scientists, but there is no any evidence how the waters are translocated upward directions. See, in the huge tree, belongs to gymnosperm tree, they are maybe growing in the 500 feet. How the water is transported to till end of the plant. Till end of the plant is water is essential for photosynthesis. How they get transported? A lot of theories are explained, but the only accepted theory, transpiration pull. If the water is lost, it creates a pull. That is why the water translocation upward direction. It's nothing but ascent of sap. So ascent of sap, absorption of water, mineral absorption. Certain minerals which is present in the soil, it is also gets enter into the root system along with the waters. If the water is entered along with the certain minerals also gets enter into the root system by passively, not for actively. Passive in the sense without utilizing any metabolic energy, they simply move from high concentration to the low concentration enter. So these are the processes are depends upon the transpiration. That is why transpiration is essential. At the same time, it is evil if there continues. Loss of water is continuous, definitely it is harmful to the plant. And apart from the transpiration, with the help of the plant, I can withstand against a scorching sunlight. A plant can withstand against of scorching means extreme sunlight due to transpirations. Transpiration to make a cooling effect of the leaf. If the transpiration does not take place, what will happen? The leaf become dry and withered. As a result, rate of photosynthesis also get arrested. So it is necessary, but at the same time it is evil. So these are the significance or importance of transpirations. The next, translocation of organic solute. Solute. What do you mean by solute? The food material which are moved in a solutions. It is a solute. Translocations of solute. Translocations are takes place from where to where. One is called translocations. The leaf synthesized organic food material, that is one side. Organic food material that should be stored in the form of a macromolecule substance is called starch. Starch is a polysaccharide. When they require this organic macromolecule starch, which is converted into simple sugars. So starch, they cannot be translocated from one part to the other part. But these macromolecule substances are converted into simple sugar, for example sucrose. Simple sugar. The simple sugar is translocated different parts of the plant body. For example, yes, the leaf, it is a site of food production. It is an actual site of photosynthesis. It is an actual site of synthesis of food material. But this site of food material is synthesized by the leaf that is stored in the form of starch here. Starch. But when they require, they convert it to sucrose. The starch, when they are mingled with water, is converted to sucrose. They are translocated to other parts where they required, for example, root. Root, they required food material for survival. Because root is a air chlorophyllus, they could not manufacture their own food material. They required food from where to where? From sources to the sink area. It is nothing but sink. Sink, it is nothing but, it is a part of the plant body from where they receive the food material for survival or for storage. These are called sink. It's a source. Leaf, it's a sources, or it's a site of food production, is a source. So from where to where, from source to sink, these are called translocations. Whether the translocation is essential? Yes. How? Because the site of food production and the site of storage and utilization, it is further apart. It's separated further apart, is a long distance. Due to long distance, translocation has to be essential for the survival of different parts of the plant, for example, root, leaf, bud, flower, fruit, seed. These are the organ for storage. It's not for the organ for synthesis. It is only for storage organ. Whereas synthesis, leaf is a synthesis part. So the process of translocations of organic food material from the region of site of production or site of synthesis to the region of storage or utilization. These are called translocation of organic solute. Now it is a question in front of you, which essentially is involved for these translocations. For this long, there are two types of translocations: short translocation or also long translocation. From leaves to root, it's a long translocation. From leaves to the shoot tip, these are the short translocations. So based on this translocation, certain transporting substrate, transporting mechanism are involved for this translocation, which is a mechanism, phloem. We know that xylem and phloem, it is a complex tissue. Xylem is mainly involved in the water conducting elements. Phloem is mainly involved in the food conducting elements. So where is the path of translocation? Translocation takes place from one side to the another side through phloem. Phloem, it is a path. How can be demonstrated? The phloem, it is mainly involved translocation of food material. How can be demonstrated? By using the experiment is called ringing experiment. It is also called girdling experiment. Ringing and also girdling experiment. The next, ringing experiment or girdling experiment. So this ringing experiment, which is used to demonstrate that translocations, it takes place by phloem. It is clearly demonstrated by ringing experiment. So in the ringing experiments are involved to remove all the tissue which is present outside the vascular cambium. Then first, what is called vascular cambium? Cambium actually is a meristematic tissues which is present in the vascular bundles such as xylem and phloem is called vascular cambium. Yes, it is a woody plant or woody stem. If we cut the woody plant, the vascular bundles are arranged in the form of ring-like structures. This type of arrangement is called U-stem. Center pith. Pith is made up of loosely arranged parenchyma cells. This ring, it is a vascular cambium. There are two types. It is a vascular bundle, outside phloem, inside xylem. Xylem, outside phloem. So both the xylem and phloem always organized together in a bundle. These are called vascular bundle. The cambium ring which is present in between the xylem and phloem, in between, these are called intravesicular cambium. Intravesicular cambium. The cambium which is present between the vascular bundle. Yes, here this between the vascular bundle. These are called intervesicular cambium. Inter, between, intra, within. So both the intravesicular cambium, intervesicular cambium, they are together to form vascular cambium. So vascular cambium, two types: intervesicular cambium, intravesicular cambium. So in this experiment, these are the totally vascular cambium. The tissue which is removed outside the vascular cambium. So it is a vascular cambium. Outside the vascular cambium, first one phloem. To remove the phloem also, yes, phloem is removed. Outside the phloem, it is a cortex. Cortex. Outside the cortex, it is a epidermis. In the woody plant, the epidermis become thick to form bark. Marapatta, bark. So all the tissues are removed outside the cambium. What are the tissues which is present outside the cambium? First one is phloem. Second cambium, cortex. Third one is bark. So all should be removed. So which one is there? Only xylem is only there in the ringing experiment. So for clear understanding, this is a potted plant. Yes, it's a ring pot or griddle pot. So to remove the bark in the form of ring, these are called ringing experiment. So in the pot, only xylem is there. The xylem only present in the ringing part. Other part outside the vascular cambium, phloem, cortex, and epidermis, bark, everything are removed. So only xylem. This xylem only connected both upper and lower part of the plant is connected by only xylem. This whole setup is kept in a beaker containing water. Beaker containing waters. This whole physical setup is kept in a bright sunlight for one day or maybe 5 hours. After regular interval of time, after 5 hours, we can observe that. There is a swelling in the upper part of the ringing area. The upper part of the ringing area that gets swollen. These are the swollen part. What will happen? Due to the accumulations of food material. The water is essential for photosynthesis. Water is there. Carbon dioxide is absorbed from atmospheres. Chlorophyll pigment is also there. Sunlight also there. So the leaf, they manufacture carbohydrate. That is a site for synthesis, food. Now the food is converted into simple sugar. They get translocated through phloem. If here the phloem is removed, so their failure to translocation of food material into the root system because of phloem is removed. But due to the absence of phloem here, the food material gets accumulated. That's why that regions which is present above the removed or ringing areas is become swollen. If these conditions are continued within a days, what will happen? Root will die first. If the phloem is removed, root will die first. Why? Root, they cannot manufacture their own food material because it is a non-chlorophyllous part. Achlorophyllous part. They could not manufacture their own food material. They derive the food from the leaf. Here the food material does not pass to the root system because phloem is removed. Next, if the xylem is removed, what will happen? Shoot will die first. It is a neat question. Because xylem is the only part for conduction of water. If the xylem is removed, they failure to transport of water to all parts of the plant body, shoot will die first. If the phloem is removed, root will die first. So from that experiment we can conclude that the phloem, it is responsible for translocations of food material to various parts of the plant body in both upward direction as well as downward directions. So these are called ringing experiment or also called curling experiment. The next, what are the direction of translocations? Direction of translocations. How translocations are takes place, in which directions? The phloem translocate the food materials from the site of production. From the site of production is nothing but leaf to the area of growth. To the area of growth such as shoot or root, flowers, to the area of growth and also development. These are called direction of translocations. So based on this category, what are the directions are, how their phloem are translocate the food material in three directions. One is downward directions. From where to where, it's a long transport from leaves to the root. From leaves to the first of all stem to the root. It is a downward directions. The second one is upward directions. Third one is lateral directions. The three directions. Then what is called downward direction? Translocation of food by phloem, downward direction. From where to where? From leaves to the stem. Stem also responsible for storage of food material. For example, sugar cane. Stem become thick and swollen due to storage of food material. Potato, underground stem that become modified to storage of food material. Stem also storage of food material. From leaves to the stem portion, from stem to the root. Root also, for example, carrot, beetroot, radish, they become thick, swollen. They perform a secondary function. In addition to the primary function, they perform secondary function, storage of food material. So these are called downward directions. And next one is upward directions. From where to where? From leaves to the neighbor part. Which is the neighbor part? For example, bud. Floral bud. They require food for development. Bud, fruit, and seed. These are required. So from leaves to the bud, fruit, seed, these are called upward directions. And another one example for upward direction, germinating seeds. Germinating seeds. If the seeds are placed under the soil, what will happen? They become imbibed, absorbs large amount of water through a minute opening called micropyle. If the water is absorbed by the seeds, they become imbibed. The seed coats are burst out. There are two types of seed coats which cover the seed. One is outer seed coats called testa, inner is called tegment. If the seed coats are burst out, what will happen? The seedlings, germinations are takes place. Seed contain two portions. One is a embryo, another one is a endosperm. The embryo that's developed a future plant. Shoot and root are developed from the embryo part. Developing embryo absorbs the food from where? Endosperm. Endosperm is nothing but is a storage of food material. It's a nutritive tissue for growing embryo. So the seed is a reservoirs of food material. If they are at the time of germination, it supplies the food to the seedlings. Seedlings grow upward direction. So this type of translocation is nothing but the upward directions. Germination of seeds, it is a very good example of upward directions. Next, lateral directions. From where to where? From the center part, pith, then to the cortex region, then cortex to the epidermis, then bark. So the food translocation from where to where? From center towards the periphery. These are called lateral direction. So the lateral direction, especially translocation food takes place through medullary ray. What do you mean by medullary ray? The pith gets extended between the vascular bundle. These are called pith ray. It is also called medullary ray, through which the food metals are translocated in a lateral direction. From pith to the cortex, cortex to the epidermis, then lateral direction. So these are the three types of directions are translocated by phloems. Then what do you mean by source and sink? It is another one question. Source and sink area. Difference between sources and sink. Source. Any part of the plant body which is capable of manufacture their own food material, manufacture or synthesis of food material, which is translocated to the other part. These are called source area. Which is an actual source area of the plant body. Leaf, it is an actual source and site of the photosynthesis. These are called sources. What is called sink area? Any part of the plant body from which where they receive the food for their survival. For example, root, stem, bud, fruit, seeds. These are does not manufacture carbohydrate, except some stems can manufacture, but maximum these part of the plant body, they could not manufacture carbohydrate or synthesis their own food material. They are responsible to receive the food material for various metabolic activities from the leaf. So these parts are called sink. This is called source to sink. So food is always translocated from where to where, source to sink. Here nothing but in the sources area, phloem is loading. Phloem is loading the food material. Sink here, phloem is unloading. Thank you. And you watch my videos continuously without skipping, you may easily understand. The next thing we are going to discuss next video.